First, I would like to thank the organizer for giving me the opportunity to present our data here. In my talk, I will briefly discuss how the HRAS gene, the Harvey RAS gene, is controlled by a mechanism involving non-bDNA conformations. Then I will discuss about the possibility to target these non-bDNA conformations in order to inhibit the HRAS gene in cancer cells. And I will report data about the effect of antrathiophenediones in cancer cells and the cellular response to this to the suppression of HRAS. The RAS gene are among the most mutated genes in human cancer. It has been estimated that about 30% of all human cancers contain mutations in the RAS gene. This gene encodes for a protein of 21 kilodaltons, which is active when it's bound to guanosine triphosphate. In the active state, it promotes the proliferation of the cells through the MEK-ERK pathway. But when guanosine triphosphate is hydrolyzed to guanosine diphosphate, the protein loses its activity. Mutations in the RAS gene results in a protein that is not able to hydrolyze GTP into GDP, so the protein remains locked into the active state that transmits constitutively to the cells signals for proliferation. And this is the mutations are the genetic lesions that contribute to the malignant transformation of the cells. For this reason, the RAS gene are very important target in chemotherapy. In my talk, I will focus on the Harvey RAS gene because mutations in this gene are associated to urinary bladder cancers. So in order to propose a novel strategy to inhibit this gene in cancer cells, we have focused on the promoter of the gene. We discovered that immediately upstream of the major transcription start sites, there are two GC-rich elements that we call HRAS1 and HRAS2. These GC elements are composed of blocks of guanine and can in principle fold into a non-bDNA conformation, which is called a quadruplex DNA. It is a folded conformation stabilized by tetrads of guanine. So to demonstrate that the GC elements can fold into a quadruplex structure, we performed dimethyl sulfide footprinting and the circular dichroism. Through this technique, it is possible to identify the guanine that are involved into the formation of the quadruplex. And the CD gives information about the topology of this folded conformation. And so I'm not going to the detail of this experiment for reason of time, but we concluded that HRAS1 folds into an antiparallel quadruplex stabilized by 3 tetrads of guanine. As for the second sequence, HRAS2, it folds into a parallel quadruplex, which is very stable. At 90 degrees, the conformation is still present. It has a melting temperature that is higher than 90 degrees. We also focus on the opposite cytosine-rich strand, and we discovered that also this strand is able to fold into a non-bDNA conformation. It folds in the so-called I-motif conformation stabilized by cytosine pairings. These conformations is stable under slight acidic conditions, but it is possible that in the presence of genome superhelicity, it can be also stable under physiological conditions. So after having demonstrated that the GC element of the RAS gene are able to fold into these non-bDNA structures, we asked if protein can can bind to these G elements. And we performed streptavidin biotin pull-down assay coupled to mass spectroscopy and also chromatin immunoprecipitation assays. And the results are here reported. We discovered that two zinc finger protein, MAZ and the SP1, MAZ means MYC-associated zinc finger protein, and SP1 recognizes the two G elements of the HRAS promoter, and also heterogeneous ribonucleoprotein I1 is able to recognize these two G-rich elements immediately upstream of the major transcription start site of the gene. We then wanted to know if this protein are able to recognize the non-bDNA conformation that can be formed by these two GC elements, and by mobility shift assay using the recombinant forms of the protein, we found that MAZ is able to recognize the two quadruplexes formed by HRAS1 and the HRAS2 respectively. Whereas we found that the pyrimidine-rich strand, which is able to fold into the I-motif conformation, is recognized quite tightly by heterogeneous ribonucleoprotein I1. You can see here the complex between the protein and the and the DNA. About the binding of MAZ to the G-quadruplexes, we found that this binding is let's say, catalytic in nature, in the sense that the protein is not only able to bind to the quadruplex, but also unfold the quadruplex. And this was demonstrated by fluorescence resonance energy transfer experiment. Here is the results of a typical experiment. The quadruple forming sequence is labeled at the 5' and the 3'. Something. Yeah, but the pointer is not. Okay, you can do that. So the quadruple forming sequence is labeled at the 5' and 3' ends with two fluorophore, fluorescein and rhodamine. When the sequence falls into a quadruplex, the two fluorophore come one close to the other, and so they can communicate each other. So when fluorescein is excited at 580 nanometer, it emits fluorescence at 520 and 580 nanometer, spectra 1 to 3, because there is a crosstalk between, there is Fret effect between the two fluorophore. But when the protein, when the DNA, the quadruplex, is incubated in the presence of the protein, we observed a dramatic increase of the fluorescence at 520 nanometer, and this is indicative of folding. This means that the end-to-end distance in the quadruplex is increases due to the unfolding effect of the protein. We also measure by we applied the Fret equations and measure the end-to-end distance in the quadruplex, which is about 40 angstrom. But when it is incubated with the protein, it increases to 58 angstrom. This is an indication of unfolding, but the unfolding was also confirmed by Fret melting experiment. In the absence of a protein, the quadruplex melts, the quadruplex structure melts with the typical sigmoidal curve. In the presence of protein, this melting profile is abrogated. You can see that the melting 4, 5, and 6 are obtained in the presence of the protein, and this demonstrates in a very clear way that the protein melts the structure. So at this point, we wanted to know what is the impact of the quadruplexes into the transcription of HRAS. So to answer this question, we constructed a vector in which luciferase was driven by the entire HRAS promoter. Then, by site-directed mutagenesis, we obtain a number of mutant vectors in which we introduced point mutations in HRAS1, HRAS2, or in both HRAS1 and HRAS2 in order to destabilize the capacity of the sequences to fold a quadruplex. In brief, again, I'm not going to the detail of these experiments, but the main result is that when the two quadruplexes are destabilized by the point mutations, but the point mutations do not interfere with the binding of the protein to the promoter, we observed a dramatic increase of the fluorescence. And these results of the luciferase, and this means that the two quadruplexes they behave like a repressor of transcription. Then, to know what is the effect of the proteins SP1, MAS, and heterogeneous ribonucleoprotein R1 on the promoter, on the expression of HRAS, we simply silenced this gene by small interference RNA, and we observed that when these genes, the genes encoding this protein, are silenced, the expression of HRAS is strongly downregulated. So, put together all this data, we propose a model which for the regulation of HRAS transcription. So under normal conditions, the 2G elements that controls the transcription of HRAS are in equilibrium between the linear and the folded conformation. In the folded conformations, the transcription is kept to a minimal level. But when transcription has to be activated, MAS is expressed by the cells, it binds to the quadruplexes, it destabilizes the quadruplexes. The promoter assumes the canonical bDNA conformation, which is recognized by the protein of the transcription machinery, and so transcription is activated. So according to this simple model, small molecules that are able to recognize the quadruplexes and stabilize the folded conformation of the promoter should inhibit the gene. So with this idea in mind, we tested many planar molecules because we think that the molecule should stack external to the external G-tetrad of the quadruplex. And so we tested many molecules, porphyrin, phthalocyanine, chlorine, but recently we focused on this type of molecules, antrathiophenediones. There is an anthraquinone core fused to ethiophene to get this chromophore. We linked to alkyl side chain ending with chloroacetamidino groups. These molecules have been synthesized by Andrei Sekotikin from the Academy of Science of Moscow. And the first thing that we have done with these molecules was to see if they are able to bind the quadruplex, and it turned out that they bind quite tightly the quadruplex. They have quite a stronger affinity for quadruplex DNA. Here are the dissociation constant, which is in the order of about 200–300 nanomolar. And this was obtained by Fret titrations experiment. Then we also observed that these molecules are able to stabilize the melting temperature, the quadruplexes, quite efficiency, because the TM increases up to 25 degrees, and the extent of stabilization is directly proportional to the length of the side chain. We also performed a competition melting experiment to know if they have a quite they have quite a good quadruplex to duplex specificity, and we could conclude by this experiment that the compounds, that this antrathiophenediones, they have more affinity for quadruplex DNA than duplex DNA. In the presence of an excess of duplex, you don't have any change in the melting temperature of the quadruplex. This suggests that they have quite a good quadruplex to duplex specificity. Then, before using these molecules to test their capacity to test the cell, the capacity to inhibit the HRAS gene in cancer cells, we wanted to know if they are taken up by the cells quite efficiently. So we performed cell cytometry experiments to evaluate the uptake of these compounds. And we discovered that these compounds are taken up quite efficiently by the cells, and the uptake is irreversibly proportional to the length of the side chain. The shorter is the side chain, and the higher is the uptake. And this was a bit surprising to us, because we thought that the uptake should increase with the hydrophobicity, lipophilicity of the molecules, but it was not the case. So we also observed that the antrathiophenediones are taken up by non-cancer cells less efficiently than cancer cells. They enter into cancer cells more efficiently, but this was not surprising, because usually cancer cells take up molecules more efficiently than non-cancer cells. And we also discovered that the uptake is higher at 30 degrees than 4 degrees, and this may think about an active transport. And indeed, we found that the transport occurs by endocytosis, because the transport is sensitive to dinosaur, which is an inhibitor of endocytosis. So this is the confocal for the internal distribution or intracellular distribution of the molecules was addressed by confocal microscopy. Here there is. Maybe I have 1 minute more for. This is okay. I don't have time to discuss these images, but we concluded that they show that they internalize into the nucleus. We found that the compound 9, 10, 11, they have quite a strong activity in a cancer cell, but not in non-cancer cells. They inhibit the metabolic activity of the cells. They inhibit, as we had hypothesized, the transcription. By quantitative real-time PCR, we determine the level of HRAS. It was a compound 9 and 11 strongly inhibit, I would say, the transcription. The protein, you can see, it's practically is extincted. There is protein is extincted. The cells, there are no protein. It's completely extinct in the cells. They also the extinction of the protein results in the arrest of colony formation. The cells do not grow. You can see combined 9 and 11, they completely block the growth of the cells. Then we wanted to know if this block of growth is due to apoptosis. This is typical experiment to demonstrate apoptosis, activation of caspase 3 and 7, cleavage of PARP1. You can see the cleavage of PARP1, strong cleavage of PARP1. This is annexin 5 propidioiodide shows that the population of the cells treated with 9 and 11 is completely in the apoptotic state. You see, completely apoptotic. And finally, we also found that the inhibition of HRAS results in the inhibition of cyclin D1. Cyclin D1 is a protein essential for the progression of the cells from the G2 phase to mitosis. And indeed, when we analyze the cell cycle, we see that we found that the cells are blocked into the G2 phase. Finally, we tested this compound in vivo. This is the result of a preliminary experiment. We are still performing an experiment. In the preliminary experiment, we just injected intratumorally compound 9, and we found, into a nude mice containing a bladder cancer xenograft. And you can see there is a strong inhibition of, it's quite a reasonable inhibition of tumor growth and the increase of the survival time. This is just a preliminary experiment, and we are performing now an experiment in which we have injected into the tail, the vein tail, the compound 9. To conclude, these are the people that have been involved in this experiment. Most of these are PhD students, and the research is financed by the Italian Association for Cancer Research. Thank you very much for your attention.